Okay, people, here we are again. Here we are on a lovely Friday. Started out kind of rainy, kind of sunny now. Life is good. When this class is over, let's go have some fun. Not together, though. I'll be fine by myself. Let's see your faces real quick. Trying to say hello here. I look at James and he smiles immediately. I can't tease that guy anymore. Okay, Farouk is still hiding. That's okay. You're doing well in this class, Farouk. You can hide as long as you want. What do we got here? 17 people out of, I think, 35. Hasn't no one heard about the free bonus points I'm giving out? I want to talk about that too. Asha Gupta, Bimalak. Come on, Bimalak. There's Asha. There she is, Bimalak. Oh, Kawame. Kawame. Where is he? Kawame. Welcome. Welcome, Rocky. Welcome, Vanessa. Vanessa's friends call her Nessa. She told me I couldn't, though. Okay, I'll take it. All right, so a couple of quick announcements here. One is there's no recording from Wednesday's class, which is probably just as well because it was a crappy class. I pulled it up to edit the beginning part out where there was a big break in the action, and something happened and I lost it all. So there's no recording from that. But I'm going to review some of it today, so you guys should be okay. The other thing is, looking at the material today, it's like we have a lot more to go, and it's a lot of really complicated stuff. But we've already done a lot of really complicated stuff. So I'm just wondering if you guys want to keep going and keep our exam on April 5th, which will cover all of Chapter 5, or if you would rather just stop now, I'll review on Monday and we'll have our exam on Wednesday. So I put a poll up in Moodle, in the first block of Moodle, it's going to be open till Sunday, so I want you to go there and vote on that. Also, if you've got some specific comments about the whole structure of everything, send me an email. I will definitely read it and take it into consideration. So there's that and that. What else? I guess that's about it. So why don't we go ahead and do today's quiz, and then we can talk about that after it's over. So today's quiz. I'm just bringing up my Moodle so that I can show you where it is, so you can find it on your own probably. So here's today's quiz, daily quiz 326. So I'm going to turn off my video and mute myself, and I'll give you another 11 minutes to do that. Later, dudes. And we are back. Let's take a look at these questions and answers. Hopefully you've all done well. Oh, another thing I wanted to announce while I was here is that someone in cell biology, I'm doing the same kind of quiz thing in cell biology, and people were complaining because it was so hard to get all the questions right in order to get that one bonus point. So what I'm going to do is collect up all your scores on this, on these quizzes. Each of them will be worth 10 points. And for every 10 points you get, you'll get one bonus point. So it'll be cumulative rather than win or lose each day. Okie doke. What do I do? What do I do? I will do this. I swear to God, it feels like I'm just here alone in this room by myself. I hope I don't say anything inappropriate. That was a possibility. Nishi's here, yay! So I got that going for me. All right, new share. This guy. Okay, question 1. Hippo signaling is active in cells that have a free apical surface. That is wrong. That is false. So Hippo, that one protein, AMAT, goes up to that apical surface, and then Hippo gets stuck on the scaffolding proteins and becomes inactive. Question 2. Matching. You match them. Use each one only once. Okay, so whenever I say use each one only once, that means one of the answers could go to 2 questions, but another answer can only go to 1 question. I'll show you what I mean. This blocks nodal signaling at the receptor level. Well, let's go. This blocks nodal signaling at the translational, this one here. This blocks nodal signal by blocking nodal itself. Now that could be either lefty or Cerberus, but I'm going to say Cerberus because I have a hunch I'm going to need that lefty again. At the receptor level, well, my God, there it is. That one's lefty. So lefty will block nodal directly and also bind to its receptor and block it that way. This activates nodal after it's released. After it's been released. Oh, this activates nodal after it's been released. That would be your convertase. And then translational level, that's got to be miRNAs. Okay. So the lefty could be either the first question or the last question, but you need Cerberus for the last question. I mean, you need lefty for the first question, so you use Cerberus for the last one. Convertase is going to turn that pronodal into nodal. And miRNAs will block things at a translational level. Oh, miRNAs. miRNAs are made as hairpin loop, and they're cleaved by dicer. Pre-mRNAs become part of a RISC. Oops. Block translation. Okay. That was pretty easy. I think that was an easy quiz, but I didn't want to quiz you on the stuff that I talked about on Wednesday because I did such a poor job on it. So, hey, Dr. Slish, could you go over the first half of Wednesday's lecture? My internet dropped, so I was a little confused. What was in the first half? I believe so. Yes. Yes, I can. Will I, though? Sorry. Yes, I will. Okay, let me bring the notes up so we can see what I talked about. Nice. You never confuse me with a nice guy. I am not a nice guy. I'm not a nice guy. Okay. I am losing it. I swear to God. Okay. Formation of, yes. Okay. So this is what I'm going to be talking about today anyway. So that's the beginning of it. Yeah. Yeah. I'm going to be talking about this today anyway, because I want to repeat it because I did such a job on it. Okay. So Esha, if this doesn't cover what you want to know, let me know and we'll do something else. Okay, So, well, we started talk. I think what's today? So Monday we talked about how the DVE is formed and how the DVE becomes the AVE, and the AVE moves up this way and ends up on this side. So this becomes anterior. Sorry. So the AVE anterior stuck on vestigial. No, anterior visceral endoderm is going to be releasing Cerberus, Dickoff, and Lefty. So that causes this part of the epiblast to become anterior because it's not going to have any BMP, nodal, or Wnt activity there. So that makes this anterior. Now this rim here, right, the most proximal part of the epiblast then is going to be the proximal rim. This is what's going to start to change and turn into the primitive streak. Remember, this is proximal, this is distal. This is where it implants, and this is hanging out in the breeze there. Okay, so this tissue here is extraembryonic ectoderm. Okay, extraembryonic ectoderm. It's going to be releasing nodals, and nodal, not here because of AVE, but nodal will cause nodal release. So there's like a feedback there that brings a lot of nodal up in this proximal rim here. Now, another thing that's going on at this point, and I've thrown this slide in from previous because I think it makes more sense here, is over here at this, which will become the posterior end, the visceral endoderm releases Wnt, and Wnt causes the production of Wnt and release of Wnt. So that's, again, another positive feedback loop here between these cells of the epiblast and these cells of the visceral endoderm. So now we've got nodals being released here and BMP, which we saw from previous slides. So this EXE is producing nodals and nodals and BMP. And now we've got Wnt being produced over here. And all these signals start to build on each other. So EXE is releasing nodal and BMP4, and the visceral endoderm is releasing Wnt, which is causing nodal and BMP4, and Wnt is causing EXE to make more Wnt. So you've got all these things coming up all at once, and they're only having effect in that proximal rim and on the posterior side because AV is blocking over here. So all these things bring up brachyury, which makes this mesoderm. And it's also going to make these things divide really fast. And as they divide, they're going, the new cells are all going towards that Wnt signal at the posterior side. So that's convergence. As they're dividing, they're going that way. And so because you're blocking at the AV, this is making this anterior, and this ends up being posterior. Now, one thing I corrected from last time was I was saying that brachyury in mesoderm causes things to move towards I said node. Duh, node doesn't come out till gastrulation's over. So we haven't even gastrulated yet. Where the hell's the node? Shut up, Slish. So really what I should have said was primitive streak. So the primitive streak is forming at that posterior end. Okay, and once the primitive streak forms and starts to move, it's going to release choroidal noggin, follistatin, and BMP activity is going to fall off. So BMP will no longer, BMP will no longer be important when that node starts to build momentum. Okay, so here we are, proximal rim. We've got the 3 different hormones going on. Cells are dividing and they're converging over here where the primitive streak is going to be. So as these cells converge, then they're going to go through, outside, and back, and anterior. So they're going to cover the outside of this cup. So remember, the outside is the inside, right? Mouse being inside out, this is going to be the ventral side. So just like in chick, they come from the dorsal side, go through the primitive streak, and end up on the ventral side. And once you get all the way down to the bottom, once that primitive streak makes it all the way to the bottom, then you've got 3 layers of tissue here now. You've got the ectoderm on the inside, mesoderm, and endoderm on the outside. And here you can see a lot of mesoderm goes anterior, right? We talked about that. I'll show it again in a minute. But these cells are coming through and anterior, like all along the way as they're going, they're coming through and anterior. But it becomes especially noticeable at the end of the primitive streak, because now it's going forward from the primitive streak. So this axial mesendoderm, which will become both mesodermal tissues and endodermal tissues, is forcing this visceral endoderm out of the way and replacing it. And so here's your anterior side. This now will become brain because you've got mesoderm underneath it, and it's going to be releasing FGF and turning this into brain. We'll talk about that today. Okay. And so once that primitive streak gets to the bottom, it becomes the node. So that's analogous to your Spearman organizer or your Henson's node, but it's just called node here in the mouse. Okay, so now you've gastrulated. Congratulations. Oh, and also we've got some of this EXE turns into extraembryonic mesoderm because it also saw those 3 hormones, the BMP, nodal, and Wnt. So it turned into extraembryonic mesoderm. Okay, so that's more or less what we talked about on Wednesday, except I took about 20 minutes to do it because I kept hemming and hawing all the way through it. But hopefully that's going to make sense to you now. And we can move forward now. What happens after this? Now, another thing I talked about Wednesday was once that node starts to regress and go back towards the posterior side, what's going on there? Okay, here. This is what I was talking about with mesodermal cells coming through, primitive streak stops, and it keeps going forward underneath the epiblast at that point. So now we're looking at the chick, and we're looking at the chick here instead of the mouse. They're the exact same thing, except the mouse is all folded up, so it makes it hard to really visualize. So we're going to switch here with with neurulation, we're going to switch and go to chick model. Okay, but it's the same thing. It's the same thing. Okay. So, of course, if I was to serve you mouse instead of chick for dinner, you would know there was a difference. But here at the developmental level, they're about the same. All right, so here's your node. Okay, so your primitive streak has just come up this way, got as anterior as it's going to go, and it stops. And now the Henson node is going to start releasing FGF. And if you remember from Monday, it's starting way back at Kohler sickle, we were getting FGF being released, but the epiblast was staying epidermal because it wasn't competent to respond to FGF. Remember that chromosomes were all tied up, and so they couldn't respond to FGF. Yeah. So once you get to the end of the primitive streak, once the end of gastrulation, then these epidermal cells become competent to respond to FGF. So you've got the node releasing FGF, and it comes up this way, and you've also got this precordial plate mesoderm, which is these cells that migrated through and went forward, and they're releasing FGF as well. And that's going to specify, this is going to make all of this thicken, so the cells start to divide and this whole layer gets thicker, and then they become specified to become neural. And it can only happen in this one brief period here, because once you specify that ectoderm to become neural, it no longer responds to FGF anymore. So FGF response in this ectoderm comes on and then goes off again. But anyway, that's the anterior structure story. That mesoderm coming through the primitive streak, going forward underneath the ectoderm, releasing FGF, and then the ectoderm becomes neuroectoderm, and it will eventually become the brain. Bottom line here is FGF is our neural inducer, and it's released by that axial mesoderm, axial meaning in the center, and the node to induce the brain to form from that overlying ectoderm. So that's our anterior structures. I know I'm saying this fast, but I said it a couple of days ago. I think I say it every time we get together. So, you know, you have any questions, just let me know. Chat me up here or something. We can stop and go slower. Okay, so here's another thing I talked about Wednesday, which I want to go slower and explain better. Excuse me. So here's those anterior structures we just talked about. And once they start to form, then once the primitive streak starts, it starts to regress. And as it regresses, it starts to make somites at the back end. So at the front end of the wave is your stem zone. And your stem zone is making and releasing FGF and Wnt. So all these cells here are dividing really quickly, and they're also releasing both of these hormones. And when these cells first are made, okay, so they're dividing, and the new cells that are being made are seeing a lot of FGF and a lot of Wnt. FGF brings up a neural fate, Wnt brings up, or FGF brings up SOX, and Wnt brings up brachyury. SOX is neural, brachyury is mesodermal. So these are neuromesodermal cells. So as they divide, they're seeing both hormones, and they're kind of somewhere in the middle because they're making both markers. And then as the node proceeds forward, these cells crawl backwards, and they go back and then in and under. And the ones in the center are right underneath the tan line, and they will become neural cells and only express SOX. The ones on the outside are then going to just express brachyury, and they will be axial and paraaxial mesoderm. Okay, so, oops, what happened there? Okay, so here's that again. These neuromesodermal cells, the ones that stay here, are right on top of the mesoderm. This tan line is mesoderm. That's going to be notochord. And your notochord is releasing FGF, and these are becoming neuro. And then you've got the ones on the outside that are going through and becoming axial mesoderm. Well, actually, this is axial mesoderm, the notochord. Paraaxial mesoderm is somites and everything lateral to the somites. So these are going to become paraaxial mesoderm. Okay, what else do I want to say? Okay, so later, I don't know if we're going to get to it today, but definitely by the next lecture, if we continue on in this manner, we'll talk about pre-Semitic mesoderm. That comes up a lot. So pre-Semitic mesoderm is all the mesodermal cells in this area. Okay, so if you remember, once they get through this spot, they differentiate into mesoderm. So in this area, they're going to be called pre-somitic mesoderm because they're not somites yet. And at this point, they're mesenchymal, which means they are loosely organized and migratory. Once they become somites, they will become stationary and differentiate. So it's a big difference. Okay, what am I saying here? Self-renewing. Okay. All right. So mesendermal cells. Oh, I see what I was doing here. So this is talking about the neural ones. So the notochord is the tan. Stem cells form neural mesoderm. Cells that stay on top become neural underneath the epidermis and the top of the notochord. Okay. So mesendermal cells crawl through and under the node, become paraaxial. So what is paraaxial mesoderm? Good question. I'm glad you asked. Here is this picture we've seen before, like way back in the first exam. Here's your neural tube and notochord. So this is axial, right in the middle. Paraaxial is on either side of the axial part. So you've got a somite, intermediary mesoderm, somatopleural mesoderm, coelom, and splanchnoplural mesoderm. Okay, so we talked about these before. Here you're actually seeing that where they're coming from. They come around the outside of that node down in and anterior and lateral to there. And this patterning from notochord over is going to be based on a BMP gradient, which we haven't talked about. And we will talk about it several lectures from now. But basically, these tissues out here are releasing BMP, And so you've got a gradient from out to in of BMP. Okay, so all I wanted to talk about here was I'll keep talking about paraaxial mesoderm, and that's what this is. This whole area here is your paraaxial mesoderm. So getting back to our signaling, so I showed you this on Wednesday too. You remember this is our wave front here where you've got the stem zone, and the stem zone cells are releasing FGF and Wnt. Back here at the back end is your somites, and your somites are releasing retinoic acid. So you've got a gradient of retinoic acid this way and a gradient of Wnt this way. And I like to overlap them so you can tell these aren't two separate things, they're just two different ways of looking at the same thing. And remember that retinoic acid inhibits FGF, and FGF inhibits retinoic acid. So you're getting a really strong gradient there by that reciprocal inhibition between those two. So FGF and Wnt make cells divide. Okay, so up here you've got FGF and Wnt, they're dividing, and they're pluripotent. Once you get up here, you've blocked that FGF and Wnt, which causes differentiation into somites, and they stop migrating. Okay, so the cells are migrating, they're coming through, they hit that retinoic acid, and they stop and they differentiate. And that's called, and we'll talk about this quite a bit in the future, mesenchymal to epithelial transition. So mesenchymal cells are those loosely organized ones that move, epithelial attached to each other, and they stop moving. MET, baby, MET. All right, so that's what I talked about on Wednesday. Hopefully it made more sense today. It made more sense to me, if not you. But the next area of intervention, and if we have a test on this coming Wednesday, I'll stop there. If you want to keep going, we'll keep going. I'll just give you a preview of that, of what's coming up next. So in the Xenopus, we just said anterior to posterior positioning is done by a Wnt gradient. Wnt high in the posterior, low in the anterior, FGF high in the posterior, low in the anterior. We didn't say more than that. But there's more to it than that, and we can see that here in the chick model. It's the same in Xenopus, but we can see it easier here in the chick model. And what I'm trying to say is, you know, your primitive streak stopped here and formed a node, or right about here, I guess. And as it's going forward with this wave, with this wave model, okay, those somites are forming behind it, but these somites aren't all the same. They're all going to be different. And we've got two different factors that are important to recognize in terms of somite formation. One is how fast is that node moving? The faster it moves, the more somites you're going to get, and the longer the backbone's going to be. So, you know, and in a really cool video by that acapello science guy that I like, he talks about Evo Devo, and he's talking about how, you know, a chick may have, I'm going to make a number up, may have 40 vertebrae, but you make that with like one or two changes in small genes, you make that node go faster, and it goes really fast, and now you've got a snake with 100 vertebrae. Okay, so the number of vertebrae you get has to do with this clock that has to do with how fast that node is moving down. Because as it moves, it makes another somite. So we'll talk that. I know that's broad in general. We'll talk more in depth about that. So that's one thing we need to know. The other thing we need to know is, as those cells are going through that pre-Semitic mesoderm phase, we've got genes turning on that are telling those cells what somite they're going to be when they finally finish in the end. So, do I say it here? No. For example, in your notes for today, somites. So, so your first 5 somites are going to be patterned while they're still kind of looking amorphous. There's nothing, you can't see anything there, but genes have turned on that those first 5 somites are going to become the back of the skull and the neck muscles. And then as that thing advances, the next somites are going to be cervical vertebrae. As it goes further, they become thoracic vertebrae. As it goes further, lumbar and tail vertebrae. Okay. And that has to do with Hox genes that are different Hox genes or combinations of Hox genes that are being turned on in that pre-Semitic mesoderm area as that node is moving. So, and in terms of the clock and how fast it moves, somites form every 90 minutes in chick, every 120 minutes in mouse, which tells you the chick's going to have more vertebrae, and every 6 hours in human. Humans can have even fewer vertebrae than chick and mouse. Hox genes activate during this period, and those Hox genes are going to give you your anterior-posterior positioning. Okay, so, and the somite formation, I just said this, but I'm going to read it again from the notes. Somite formation requires differentiation, which is a mesenchymal to epithelial transition. MTE, I think it may be MET, mesenchymal transition. I have to look that up. I don't know if it's MTE or MET, but it's basically changing from mesenchymal to epithelial. MET occurs at the anterior side of the node. Okay, right? The node's going forward. The back end of that wave is the anterior side. That's where it's happening. And it's happening where retinoic acid is high and FGF-Wnt are low. And the segmentation, so you don't, like your notochord is one solid thing, and somites are blocks, chunks, right? And that's because of another signaling pathway. I know you were hoping for another signaling pathway on this exam, and it's called the Notch signaling pathway. Notch is kind of interesting. It is probably the strangest signaling pathway that I know of. It's not complicated necessarily, but it's just weird for a couple of reasons. Okay, Notch. So here is your transition from pre-Semitic mesoderm into somite. And you get this like cup formation in not all species, but definitely chick and mouse, where one comes down into the other one. So what's going on right here and right here that causes that somite to form is Notch signaling. Notch is going to turn on different genes, and that's going to cause it to separate from the cells next to it and form a coherent bunch of tissue. So here's Notch. Notch is a receptor. Okay, so it's got a big extracellular domain, a single transmembrane domain, and an intracellular domain. And here are some genes that Notch would turn on, but it's got repressors on it, so it's not started now. So this is when there's no activation of the Notch receptor. The ligand for the Notch receptor is called delta. And delta is also a transmembrane protein. It's on a different cell. So this is what we call juxtacrine signaling, where one cell touches another cell and activates it. In cell biology, we talked about this. We'll talk about it more, where we talk about apoptosis and natural killer cells, will come down and touch a cell and tell it to die. Juxtacrine. Same thing here. You've got this cell touches this cell, and it's got this delta on it, which is the ligand for the Notch receptor. So that activates the Notch receptor, and it cleaves its tail off. Okay, kind of like one of those geckos. You pull on and the tail comes off. So the tail comes off, and when it does, it's a transcription factor. What? You're right. It's crazy. So it doesn't activate a transcription factor. It is a transcription factor. So when it gets active, that comes off. It goes in, it knocks the repressor off, and boom, you get those genes to be turned on. So let's see, what are the genes that get turned on? Oops. Notch is going to turn on hairy and Ephrin A4, and Ephrin A4 is going to turn that tissue into a somite. We'll get into that in the next class. Okay, so how do we know? Okay, so that's how somites get formed. Now, how do we know that the somites are patterned already, even when they're in the pre-Semitic stage? So you would perhaps if you were like me, you would fully accept that a fully formed somite already knows where it is. It already has its positional information, and it is patterned to be cervical or whatever. But over here, these amorphous pre-Semitic cells are just kind of crawling mesenchymal cells, right? And so they don't seem to look like anything, but they have been patterned already. And you can tell that by taking two embryos, you cut this section of pre-Semitic mesoderm out of one, you flip it over and stick it in the other one. So this basically just went walk, stuck it in. You stick it in backwards, and the somites are already upside, the somites are upside down, right? And if you let that embryo grow, that part of its body will be upside down. That's how you can tell that still already in that pre-Semitic mesoderm, you've already got some, got the pattern laid down. Immersion. Hang on. Here's an even freakier experiment, which I'll talk about again Monday if we continue in this vein. Here's an older and a younger embryo. So you take something from the thorax region, the pre-Semitic mesoderm that will become thorax, take it out of one embryo, and you put it into another embryo in the cervical region. What's going to happen, Dr. Frankenstein? You get cervical tissue in the neck, okay? Again, just another experiment that shows you that there's patterning done in that pre-Semitic mesoderm. So the pattern is laid down there before the somite gets formed. And that's what I wanted to talk about today. I'll talk about this again next time, and I will talk about this next time too. This is the clock that decides how fast those somites are going to be formed. Okay. So that is what I had hoped to get through today. Is everyone happy with that? Is anyone happy with that? Can I get a thumbs up? Can I get a thumbs up? No? No one's going to give me a thumbs up. Oh, I get some thumbs up. Okay, good. Kwame, you're my man. I know I can count on Kwame. Nishi says, I'm happy. As long as Nishi's happy, we're all good. Okay, guys, go ahead and vote in that poll, like I said. And if you have any specific comments, just send me an email. We'll discuss it. All right, have a great day, buddy. Bye. I mean, guys, we'll see you. Have a great weekend.